Good morning and welcome you all. So in this today's video lecture we're going to see different foodborne diseases or foodborne infection. So this topic is actually from fourth unit, but I have decided to take it first because this is pretty much related to previous lectures. So in previous lectures we have seen different types of microorganisms, they are present in role and their role in pathogenicity. So in continuation with that lectures I decided to start these foodborne diseases. So in this today's lecture we'll see how microbes cause diseases through food and their role and the mode of infection, and we'll cover lots of details regarding foodborne diseases. So let's start. So first, what is meant by foodborne diseases? So foodborne diseases, they are abbreviated as FDB, are acute illness associated with recent consumption of food. So there are two types of illnesses you can see, one is acute and other is chronic. So chronic persists for some longer time or longer period, and acute it persists for shorter time. Okay. So foodborne diseases are acute illnesses associated with recent consumption of food. So basically the illness is caused by consumption of food that is contaminated with either microbes or their microbial products like toxins. Okay. So the food involved is usually contaminated with disease, pathogen or toxicant. Okay. So why we get ill after consuming this type of food? Because this food contains pathogenic microorganisms or their products called toxicants in the form of toxic chemical compounds and other things. So such a food contains enough pathogens or toxicants necessary to make person sick. Okay. So why some people or one of the people they develop foodborne diseases? Because the load, microbial load which is necessary to cause illness or disease is sufficient. Okay. So such a food contains enough pathogens or toxicants necessary to make a person sick. Then foods, they are classified on the basis of foodborne diseases. So foodborne diseases, they are classified in different types. So the foodborne diseases are classified into foodborne infections and foodborne intoxications. Okay. So foodborne infections means these are infections that are caused due to pathogenic microorganisms which are present in food. And in case of foodborne intoxicants, so these are illness or diseases that are caused by microbial derived product, say for example toxin and other chemical compounds. So foodborne diseases are classified into foodborne infections and foodborne intoxications. Okay. So foodborne infections are caused by entrance of pathogenic microorganism contaminating the food into body and the reaction of body tissue to their presence. So what is meant by foodborne infection? In case of first type, so when pathogenic microorganisms contaminate food and that food when enters into your body, so that pathogen cause illnesses or damages to your body. So this is first type of foodborne infection. So these can either be fungal, bacterial, viral or parasitic. So the pathogen may be, it may be fungi, it may be bacteria, it may be virus, or it may be parasite. Okay. So foodborne infections tend to have long incubation period and are usually characterized by fever. Okay. So the first symptom that appears after foodborne infection is fever. So incubation period for this pathogenic organism is somewhat longer, and the main symptom is fever, and it takes almost few days to take actual infections. Okay. So foodborne infections includes cholera, salmonellosis, typhoid fever, shigellosis, then yersiniosis, then E. coli infection, then campylobacteriosis, then Vibrio parahemolyticus and listeriosis. Okay, so these are different types of foodborne infections. In that, first is bacterial foodborne infections. It includes cholera, salmonellosis, then typhoid fever, then shigellosis, then yersiniosis, then Escherichia coli infections, campylobacteriosis, then Vibrio parahemolyticus and listeriosis. Then next type is mycotic foodborne infection, means these are foodborne infections that are caused by different types of fungi, and they are categorized or classified under mycotic foodborne infections. It includes Candida species, then Sporotrichs species, then Vangela species, etc. Then there are some viral foodborne infections also. It includes hepatitis A and Novak virus and poliomyelitis virus. So this is the overall scenario of foodborne infections. Okay. So first we'll in this lecture we're going to cover the details of all these foodborne infections in step by step or one by one manner. So in that, first we're going to study salmonellosis. It is again important group of foodborne infection or important foodborne disease. The salmonella constitute a group of organisms with over 2000 different serotypes. Okay, so this group almost contain 2000 different serotypes. So these organisms are capable of causing diseases in animals and man when taken into body in sufficient numbers. So many salmonella species have a wide host range, and these are organisms which commonly cause food poisoning. Okay. So the disease caused by salmonella species is known as salmonellosis. However, some are restricted to a single host species. Example, Salmonella abortus ovis causing abortion in eaves, and Salmonella gallinarum causes fowl typhoid. Conversely, some Salmonella serotypes are associated with human diseases and are not known to affect animals. So this particular statement shows host specificity, that particular Salmonella bacteria it affects or infects particular host, and in these two lines you can show host specificity. Okay, so these are some examples. Say for example, Salmonella typhi and Salmonella paratyphi. Okay, so these are bacteria that affects humans, and these are based human diseases. Okay, so Salmonella are ubiquitous in gut of human and animals and acts as a source of food contamination. So these bacteria basically present in gut of humans and animals, and you can say that this is a part of normal body flora. Okay. So people who are carriers of salmonella contaminated food. Okay. So heavy dose up to 10,000 or up to 10 lakh organism per gram of food is required to cause infection. So this is a bacterial load that is required to cause infection. So the dose means number of microorganisms. Okay. So in case of any diseases or pathogenic disease, so microbial load is very necessary. One, two, a few of the cells they cannot cause infections. Okay, they are eliminated by our defense system. But if microbial load is high, then there are mostly the chances of occurring of infection and illnesses. Okay, so the heavy dose up to 10,000 or 10 lakh organism per gram of food is required to cause infection. So Salmonella grow well on food and can exist for considerable period in fecus and on pastures. Okay. So common food poisoning serotypes. So these are some serotypes, different types of bacteria, and these are common food poisoning organisms of Salmonella group, and they includes Salmonella typhimurium, then Salmonella enterohemorrhagic, then Salmonella Dublin, Salmonella soft bug, then Salmonella virchow, then Salmonella Montevideo, Salmonella infantis, and Salmonella Newport. So these are some serotypes of this group Salmonella, and these are associated with food poisoning. So these species are also involved in causing diarrhea in animals. Okay. Now heat resistance. So Salmonella are killed by temperatures attained in commercial pasteurization. Okay, so this is very good thing that Salmonella are killed by temperatures attained in commercial pasteurization, and they can remain alive in moist earth for 1 year and dry earth for 16 months. Okay, so they are not destroyed in carcasses or offal maintained in chilling or freezing temperatures or in usual pickling solutions. Okay. So these are some outbreaks caused by due to Salmonella food poisoning, and outbreaks occur in 4 different forms. Sporadic cases involving only 1 or 2 persons in a household, then family outbreak in which several members of family they are affected, then large outbreaks they are caused by widely distributed infective food items, and institutional outbreak which may be caused by contaminated single food item. Okay, so these are Salmonella food poisoning outbreaks. And factors associated with Salmonella food poisoning outbreaks. So consumption of inadequately cooked or thawed meat or poultry, then cross contamination of food from infected handlers, and presence of flies, cockroaches, rats in food environment and acts as a vectors of diseases. So these are basically the sources through which these Salmonella bacteria it enters into food, and these are some sources and from which there are chances of occurring of salmonellosis is much more. Okay. Then transmission of diseases. So Salmonella reach food in many different ways. So directly from slaughter animal into the food, and from human excreta, and transferred to food through hands, utensils, equipments, flies, etc. So these are again transmission means sources through which this Salmonella bacteria it enters into the food. So first it can directly enter from slaughter into animals to food, then it can enter through human excreta and transferred to food through hands, utensils, equipments, flies which are contaminated with these organisms. Then food poisoning is more likely to occur if a total number of microorganisms present in high. A smaller number may have no ill effects. As I have told you before, a sufficient microbial load is necessary in case to occur any infection or any illness in body. Okay. Then which type of foods involved? So any food contaminated with Salmonella may be involved in this Salmonella illness or Salmonella disease, Salmonellosis disease. However, food commonly involved are animal derived foods such as meat and meat products, then milk as well as milk product, and egg as well as egg product. So basically we'll get this infection, it is derived from animals. So it is these are products that are totally derived from animals, say for example meat and meat products, then milk we get from animals, then eggs we get from animals. So animal derived product are more susceptible to and there are more chances to cause salmonellosis after consuming these animal derived products. Then clinical symptoms. So the ordinary symptoms includes abdominal pain, headache, then diarrhea, then fever, then vomiting, then prostration and malaise. So these are basic symptoms associated with these salmonellosis diseases. And in severe cases there is septicemia with leukopenia, endocarditis, then pericarditis. So these are technical terms associated with medicine or medical. These are medical terminologies, and these are basic symptoms as well as in severe cases their complications may occur in more large, or what we can say that in a very severe cases there is a septicemia with leukopenia, endocarditis and pericarditis. So these are severe medical health aspects. and severe cases are encountered in babies, young children, the sick and elderly person. So the mortality is up to 13%. So basically this disease is related to babies and young children due to their poor immune system or poor immune status, and in case of sick and elderly person also their immune system is not so stronger, so to fight these diseases or to eliminate this disease. Then control measures, how to we can control this disease. So efficient refrigeration and hygienic handling of food. So these are the two main. Hygiene is important in all diseases or in removal of all case of pathogens. So hygiene and freezing is very much important. Not in this case also, but in other diseases also. So consumption of properly cooked meat. Then complete thawing of frozen meat and adequate cooking, and heat processing of milk, then milk, fish and poultry, and to destroy salmonella organisms in food. So these are control measures that by using these control measures we can minimize the chances of infection is caused by salmonella. The next disease is typhoid and paratyphoid fever. Again, it is a very important disease as far as foodborne illness is concerned. These are also known as enteric fevers, and enteric fevers includes typhoid and paratyphoid fevers caused by Salmonella typhi and Salmonella paratyphi A, B, C respectively. So these three types of groups of Salmonella, they cause these illness or the illnesses and infections. So the serotypes are similar to other Salmonella bacteria, but unlike them, they are essentially parasites of man. So Salmonella typhi possesses a capsular antigen in addition to usual O and AH antigen found in other serotypes. So this property we can use for diagnosis of Salmonella, typhoid and paratyphoid infections. So disease symptoms. So the incubation period is usually 2 weeks, but it might vary from 3 to 28 days for typhoid fever and between 1 to 15 days for paratyphoid fever. The enteric fevers, they are generalized septicemic infection with frequent if not constant bacteremia during first 2 weeks of disease. Then abdominal symptoms are severe, while fever and illnesses may cause 4 to 6 weeks. So these are overall symptoms and incubation period for this Salmonella typhoid as well as paratyphoid diseases. Then transmission of disease occurs. The typhoid and paratyphoid bacilli are essentially human parasites and are required mostly from human sources, namely patients and carriers. So this type of infection is mostly caused by human to human, and the bacteria can be transmitted by contamination of water, then milk or fluid by flies. So these are some sources through which this bacteria can enter into your body, and only few organisms needed to cause diseases. So here in case of this particular disease, so microbial load is low as far as Salmonella is concerned. So very few bacteria, they can cause infections in our body. Then control measures. So hygienic control of food and water supplies, then detection and treatment of chronic carriers, and vaccination using TAB vaccine. The vaccine contains mixed culture of Salmonella typhi and paratyphi. The vaccine protects up to 5 to 7 years. So overall these are control measures. So you have to keep hygienic conditions of food as well as water. Then you should detect and treat the chronic carrier. So those who are carriers for disease, they should be isolated and they treated. And there is vaccine which is popularly known as TAB vaccine, which is used for young children, and it gives it can protect the children of age group up to 5 to 7 years. And the next disease is campylobacteriosis. So campylobacter are group of tiny stickly micro aerophilic curved or spiral gram negative rods. So campylobacter, these are a group of bacteria having tiny strictly microaerophilic curve. So these are strictly microaerophilic. Then these are curve shape or spiral gram negative rods, means these are rod-shaped bacteria having curve or spiral shape, and these are always gram negative. So Campylobacter jejuni and Campylobacter coli, they are the main bacteria of this group. They causes campylobacteriosis, and Campylobacter jejuni and Campylobacter coli cause food poisoning and are associated with acute enterocolitis in man. So Campylobacter jejuni occur in large number in cattle, ficus, and poultry as a normal flora. So this bacteria, again it is related with animals, and Campylobacter coli are commonly associated with human diarrhea and enteritis in pigs, and mostly in association with Trypanosoma hyodystenory. Okay. So this is overall information about campylobacteriosis. It is caused by Campylobacter jejuni and Campylobacter coli, and in case of diseases in man, so Campylobacter jejuni and Campylobacter coli causes illness characterized by diarrhea, then abdominal pain, then fever, then nausea, vomiting, and abdominal complaints. So these are overall basic symptoms, or different types of symptoms after entry of or illnesses caused due to Campylobacter jejuni and Campylobacter coli, and these are common symptoms you can see in case of these diseases. It includes diarrhea, abdominal pain, fever, nausea, vomiting, abdominal complaints. So one thing keep in mind that these are common infections or common symptoms. These can appear by several infections due to several pathogens. So you cannot judge which type of pathogen causing these infections, as these are general symptoms caused due to entry of pathogen. So you can detect only after performing several tests, by after culturing or some blood test. So you cannot judge that by observing these common diseases. So the jejunum, ileum, and colon are primarily affected, resulting in acute inflammation and occasionally abscesses formation. So these are sites of actual illnesses that are actually these are parts of our digestive tract. In that the jejunum, ileum, and colon are primarily affected, resulting in acute inflammation and occasionally abscess formation. So this disease is self-limiting. These are clinical signs. So incubation period ranges from between 2 to 11 days. So it takes almost 2 to 10 or 11 days to incubate or to show symptoms after infection with this Campylobacter bacteria, and it is preceded by fever followed by foul smelling and watery diarrhea, which runs for 3 to 4 days, and the diarrhea may sometimes contain blood and mucus in fecus. So this is again peculiar thing in case of campylobacteriosis, that diarrhea may sometimes contain blood and mucus in fecus, though abdominal pain is associated with backache and high mortality. So the condition is self-limiting but may last for up to 10 days. So these are our clinical signs associated with campylobacteriosis. And mode of infection. So in case of mode of infection, so infections occur by ingestion of Campylobacter organism in contaminated food stuff. So foods that are contaminated with Campylobacter bacteria or organism leads to cause this disease, and foods involved includes meat from infected animals, then unpasteurized milk, and possibly cross contamination from these sources to food eaten uncooked or unrefrigerated. So among the meat, poultry constitute the greatest potential source of infection to humans. So basically or mostly this disease is transferred through poultry products and poultry goods. Then microorganisms are present in poultry gut and fecus up to 10 lakh organism per gram of ficus. So this is microbial load. So carelessness in kitchen, example cutting chickens and same knife used to cut other foods without proper cleaning prior to use. Then pork is major source of Campylobacter coli. So contamination of pork occurs during slaughter. So all these are sources that are responsible for causing these diseases. And in case poultry is a predominant factor in case of this disease. Then preventive measures. So through cooking of all foodstuff derived from animal sources. So this is not in case of this disease, but in case of other disease also, that you should cook your food in very well manner, so to avoid or to eradicate pathogens and to avoid infection. Then prevention of recontamination after cooking. Then proper refrigeration of foods and recognition, control and prevention of Campylobacter infections in animals and maintenance of high standards of hygiene. So these are different preventive measures that you can take to avoid this particular disease. The next infection is Escherichia coli foodborne infection. Again, this is a very common infection found in the category of foodborne infection, and E. coli are potential food poisoning pathogens which are widely distributed in low numbers in food environment, and E. coli strains involved in foodborne infections fall into the following groups. So these are different types of E. coli, and it includes enteropathogenic E. coli, that is abbreviated as EPEC. Then enterotoxigenic E. coli, it is abbreviated as ETEC, and enteroinvasive E. coli, EIEC, and enterohemorrhagic E. coli, EHEC. So these are different strains of E. coli that are involved in several foodborne illnesses or foodborne infections. Okay. So E. coli foodborne infection in general, each group is composed of unique O and AH serotypes. So these are serotypes based on different these antigens that are O and H antigens, and each group possess virulence factors which is characteristic to that of particular group. So each group has different virulence factors, and serotypes they are characterized by using O somatic and H flagellar antigens. So these serotypes we have seen previously in our previous slide. So these are basically categorized, and you can diagnose these serotypes based on these O somatic as well as EH flagellar antigens. So O and EH antigens are important in case of E. coli diagnosis or E. coli classification is concerned. So first in that we are going to see enteroinvasive E. coli. So it is EIEC strains cause illnesses that is characterized by watery diarrhea in most patients. Then in addition there is fever, nausea and abdominal cramps. Then bloody diarrhea may occur in fewer than 10% of patients, and the presence of mucus and polymorphonuclear leukocytes in stool is a typical of these strains. And illness is usually self-limiting, lasting for 2 to 3 days. So enteroinvasive means this particular group of E. coli, it invases into your body parts, especially in your stomach. So after invading into your body, it causes tissue damage and damage of cells. So these are basic features of enteroinvasive E. coli. So we'll continue. So a relatively high dose, that is 10^8 cells is necessary to produce diseases in volunteers. So this is a microbial load, and the median incubation period is 18 hours. It ranges from 2 to 48 hours. Then foodborne infections spread is a usual mode of transmission. So this enteroinvasive E. coli is mostly transferred into human body through contamination of foods, and person to person spread has been also reported. So This is a first type enteroinvasive E. coli, and next is enterohemorrhagic E. coli. So EC infection is caused by Escherichia coli serotype that is numbered, and it causes hemorrhagic colitis in humans that is characterized by diarrhea, abdominal pain, which may be severe, and in case of vomiting, and few patients develop fever. Then this illness lasts for 4 to 8 days, although it may extend for up to 13 days for severe cases. So patients with complications have bloody diarrhea, acute ulcerative and ischemic colitis, and submucosal edema, and severe chronic colonic inflammation. So enterohemorrhagic means it causes loss of blood, and so the organism E. coli, this is O157S7 is a strain, number of strain, and it is heat sensitive but resistance to freezing. It goes poorly at 45 to 40 degrees Celsius with no growth at 45 to 5 degrees Celsius, suggesting that in presence it may not be detected by fecal coliform assay test. Death occur in patients who develop hemolytic syndrome, and HUS can be recognized by acute renal failure, then microangiopathic hemolytic anemia, and thrombocytopenia. So all these are clinical complications associated with this enterohemorrhagic E. coli. Then control measures, so proper cooking of hamburger and other meats, then avoid cross contamination of foods in kitchen, and good personal hygiene. So these are control measures that you can use to avoid or control these diseases. So these are diseases. So there are lots of diseases are remaining, and we'll see remaining diseases in upcoming lectures. So thanks